Hello everyone and welcome to our module on bacteria. So let's start by talking about two general types of organisms. Prokaryotes are a very old form of life on the planet. They were probably the first form of life to evolve. And prokaryotes have no membrane-bound organelles and they have no nucleus, so the nuclear material just sits freely inside the cell in the cytoplasm. And you should know that bacteria are prokaryotes. On the other hand, eukaryotes are a more modern form of life. These are cells that have membrane-bound organelles. They do have a nucleus with nuclear material inside. And examples of eukaryotes are plant and animal cells, so the cells in the human body are eukaryotes. Also protozoa, which cause infection. We'll talk about that in another module. And fungi, which cause infections. But it's very important that you understand that bacteria are unique in that they are members of the prokaryotes and that they are a very old form of life on the planet, very different from eukaryotic cells. So now let's talk about bacteria in a little more detail. So bacteria are single-cell organisms, and one of the most important portions of the cell, especially from a medicine point of view, is the cell wall. For the bacteria, the cell wall protects the organism. It gives general support to the bacteria. It also, very importantly, provides protection from osmotic pressure. If not for the cell wall, electrolytes could get inside and swell the cell. But from a medical point of view, we care a lot about the cell wall because it's a target for the immune system, it's a target for antibiotics, and the cell wall is one of the most important parts for differentiating bacteria into the different types. So in another module, I'm going to talk in detail about the different stains we use to identify bacteria, but the most important stain we use is called the Gram stain. And this classifies bacteria as either gram-positive or gram-negative bugs. And if you look at this slide right here, you can see that there are deep purple circles, and those are bacteria that are gram-positive. They have taken up the Gram stain, and thus they have become dark and purple. The red organisms on this slide, like the ones I'm circling here, have not taken up the Gram stain, and therefore they look red or pink on the slide. So using the Gram stain to separate bugs into gram-positive and gram-negative is one of the most important identification methods we use. And the reason some bacteria are gram-positive and some are gram-negative is because of the characteristics of their cell walls. So a fundamental point you need to appreciate is that the cell walls of gram-positive and gram-negative bacteria are different. On the left side of the screen, I've drawn a picture of a gram-positive bacterial cell wall, and on the right, I've drawn a gram-negative one. So both types of bacteria have a cell membrane, and we'll talk a little bit more about that in a minute. But notice that the cell wall of gram-positive bacteria is much thicker, and that's what causes it to take up that Gram stain. Also, gram-positive bacteria have molecules called lipoteichoic acids, which help them to adhere to structures. We'll talk more about that in a minute. On the other hand, gram-negative bacteria have a much thinner cell wall, and they also have what's called periplasm. We'll talk more about that in a minute, but periplasm is material in the cell wall that contains enzymes and other molecules. And then finally, gram-negative bacteria have an outer membrane. The outer membrane is unique to gram-negative bacteria, and we'll talk about the characteristics of that in a minute. So before we go any further discussing the cell walls of gram-positive and gram-negative bacteria, we need to talk about a very important substance called peptidoglycan. This is a major structural component of all bacterial cell walls, and it's a polymer made up of sheets of sugars and peptides arranged in a very specific order, and then sheets are cross-linked to other sheets so that you can get layers of sheets upon sheets of peptidoglycans. So there are two sugars that are used to create these sheets of peptidoglycan. The first sugar is called N-acetylglucosamine, or NAG, and the second one is called N-acetylmuramic acid, or NAM, and we'll talk more about how these are arranged in a minute. The peptides are always attached to the NAM molecules, and it's high yield to remember that for your boards. They are not attached to the acetylglucosamine molecules. And there are about 3 to 5 amino acids in the peptides, and these connect the sugars together. I'll show you a picture of that in a second. So these sugar peptide backbones make chains, and then the chains are cross-linked by peptide cross bridges. And you should know that peptidoglycan is the site of action of some important antibiotics. There's no peptidoglycan in the human body, so if we develop drugs that target it, they will kill only the bacteria and not affect the human cells. So penicillins and cephalosporins act on the peptidoglycan in the bacterial cell walls. So here is a schematic representation of the cell wall of a gram-positive bacteria. So first you've got the lipid bilayer, that's the cell membrane. Remember I told you that both gram-positive and gram-negative bacteria have a cell membrane. And then we've got these layers of sheets of peptidoglycan. So the sugars are shown by the pink and blue dots. So the pink dots are the N-acetylglucosamine sugars, and the blue dots are the N-acetylmuramic acid. And then there are these peptide chains, and you'll notice that the peptide chains are only connected to the blue dots, and that's because the peptide chains attach to the muramic acid sugars, not to the glucosamine. And so we've got sheets that are cross-linked by these peptide chains and sugar molecules that are cross-linked, and then we get layer upon layer of peptidoglycans making up the cell wall. So recall that I told you that gram-positive bacteria have much thicker cell walls than gram-negative bacteria. And the reason for that is because gram-positive bacteria have up to 40 sheets of peptidoglycan. Peptidoglycan represents about 50% or more of the cell wall for gram-positive bugs. Gram-negative bacteria, on the other hand, have very few sheets. Only about 5 to 10% of their cell wall is represented by peptidoglycan. And it's this thick layer of peptidoglycan sheets in the gram-positive bugs that allows them to retain the gram stain. And this is what makes them purple. And this is a very high-yield concept for you to know, especially for step 1. The majority of bacteria that cause infections in humans are either gram-positive or gram-negative bacteria, but there are a couple of bacteria that have unique cell walls that you should know about. The first one is Mycoplasma, which can cause interstitial pneumonia. Mycoplasma has no cell wall, so it's not going to gram stain at all because it doesn't have anything to take up that gram stain. It has a unique cell membrane, and it has substances called sterols, which give it extra stability so that even though it doesn't have the peptidoglycan cell wall, it still is a stable structure. Mycobacteria are bacteria like tuberculosis or Mycobacteria leprae, which causes leprosy, and these bacteria have cell walls with mycolic acid. That's why they're called mycobacteria. They don't gram stain very well, and we use special stains like the Zeal-Nielsen stain in order to identify these bugs, and that's because they have unique cell walls that aren't going to take up the gram stain. And then finally, chlamydia, which can cause sexually transmitted disease and some other infections, lacks muramic acid in its cell wall, so it also does not gram stain very well and has a unique structure for its cell wall. So everything I've talked about up to now has been about the cell wall, but if you recall those pictures I showed you a few slides ago, inside the cell wall, bacteria have a cell membrane, and both gram-positive and gram-negative bacteria have a cell membrane. And the structure of the cell membrane is a lipoprotein bilayer. It looks like this picture here. You probably learned about this in a biology class in college. There's a polar head on the outside, and there are lipids on the inside. So what does the cell membrane do for the bacteria? Well, first of all, it provides some structural support, but it's very important that you know that electron transport and oxidative phosphorylation occur in the cell membrane. These are the biochemical processes that generate ATP. And you may know that in human cells and eukaryotic cells, this is done in the mitochondria, but bacteria don't have mitochondria, and they conduct these things in the cell membrane to generate the ATP they need to survive. There are also some enzymes found in the cell membrane, and also carrier molecules that bring important substances into and out of the cell. So we talked about how the cell walls of gram-positive and gram-negative bacteria are different, but now let's talk about some of the other differences between those two types of organisms. So a unique feature present only on gram-positive bacteria are molecules called lipoteichoic acids, or LTAs. And these are a major surface antigen that drives the immune reaction to infection by these bacteria. So in research laboratories, they've taken LTAs and injected them into animals, and they've shown that these molecules can trigger arthritis and inflammation in the eye and inflammation of the meninges, and they can even trigger a cascade resulting in septic shock and multi-organ failure. They definitely induce cytokine release, and they also can bind antibodies, which will then activate the complement cascade. So when you're talking about gram-positive bacterial infections, you should know that one of the major factors driving the immune response is the presence of these LTAs on the surface. Gram-negative bacteria have two unique features that you need to know about. The first one is the presence of periplasm, and this is the space between the cell membrane and the outer membrane. So let's look at this diagram down here. We've got the inner membrane right here. Remember, that's present in both gram-positive and gram-negative bacteria, and the outer membrane here. But in between those two, we have the periplasmic space. Now, it contains some sheets of peptidoglycan, but remember, not as many as are found in gram-positive bacteria. And then it also contains many enzymes, and one of the most important types of enzymes that are found in the periplasmic space are beta-lactamase enzymes. These are enzymes that can inactivate beta-lactam antibiotics. So we'll talk more about that in the modules on antibiotics, but what you should know right here is that this periplasm is the space that holds many of the enzymes that allow gram-negative bacteria to resist antibiotics that have a beta-lactam structure. The second unique feature of gram-negative bacteria you need to know about is the presence of an outer membrane. Remember that only gram-negative bacteria have an outer membrane. And the most important component of that outer membrane is the presence of molecules called lipopolysaccharides, or LPS. These are a major trigger for the immune system in gram-negative bacteria, and these are the reason why patients who get gram-negative bacteremia get very, very sick, often much sicker than gram-positive bacteremia. It's because of these lipopolysaccharide molecules. If you look at this diagram right here, the outer membrane is right here, and on the outside there are these LPS molecules sticking out of the cell wall, and these are the molecules that can trigger a vigorous immune response. So let's talk about the lipopolysaccharide molecules in a little more detail. These molecules have three components. They have a polysaccharide component, something called lipid A, and something called the O antigen. Lipid A is the most important part in terms of the immune reaction in making patients sick. Lipid A is highly toxic and it triggers cytokine release in the body. So if you see on your boards a description of a patient with gram-negative infection who is very sick and they ask you why is the patient so sick, lipid A is probably the reason. It is that component of the lipid polysaccharide molecule that triggers the immune response. The final component is called the O antigen, and this is a target for antibodies when the body tries to fight off the infection. So let me just pause here and make a couple key points that you should remember. There are different major surface antigens that trigger the immune system in gram-positive and gram-negative bacteria. In gram-positive bacteria, it's the cell wall and membrane, and it's the presence of those lipoteichoic acids, which are unique to gram-positive bacteria. In gram-negatives, it's the outer membrane and the presence of lipopolysaccharides that trigger the immune response. Make sure you know these two things, especially for step 1. Now let's talk about capsules. So outside the cell wall of gram-positive bacteria, you sometimes find a capsule. Not all bacteria have them, but some of them do. And also outside the outer membrane of some gram-negatives, you will also find a capsule. Capsules are a sticky gelatinous layer that's secreted by the bacteria, and because it's sticky, it helps it to attach to host cells. Capsules also provide some protection against phagocytosis so that the immune system can't kill the bacteria. And for most bacteria, capsules are made up of polysaccharides with a little bit of water. There's just one exception to that rule, and that's Bacillus anthracus, which causes anthrax. It has a peptide capsule. This is very unique. It's made up of a protein called D-glutamate, and this is a major virulence factor, and it's part of the reason why patients who get anthrax can get so sick. This capsule allows for unimpeded growth. And make sure you know that anthrax has a unique capsule because it often comes up on the step 1 exam. There's a lab test called the Quelling reaction that used to be done decades ago. It's largely historical. People don't do it much anymore in the modern era. And this was used to determine whether bacteria had a capsule or not. The main use was to determine whether strep pneumonia was the cause of a bacterial lung infection. The way you do a Quelling reaction is you take rabbit antiserum and you add it to a slide that has the bacteria on it. And if you look under the microscope, bacteria that have a capsule will swell when the Quelling reaction is performed. So a positive Quelling reaction is something you would see in bugs that have a capsule. So like I said, the most important was strep pneumonia, but also all these bacteria here also have capsules, and so they would demonstrate a positive Quelling reaction if you did this test. It's important that you understand the immunology of how the body defends itself against encapsulated bacteria. Because the capsules make the bacteria resistant to phagocytosis, the body has evolved several mechanisms to overcome those capsules. The first mechanism is that B cells secrete capsular antibodies, especially IgG, and these tag the bacteria for phagocytosis. The antibodies bind to the capsule, and then phagocytes can recognize the antibodies via their Fc receptors, and therefore they can consume the bacteria. In addition, the antibodies that bind to the capsule can activate the complement cascade. This can lead to formation of the membrane attack complex, which leads to cell death. In addition, the C3b protein, which is an opsonin, can form on the surface of the capsules, and this can draw in phagocytes to destroy the bacteria. Here's a picture just to make this a little more clear and to help you remember it. So we have a bacteria right here, and a couple of IgG antibodies have bound, and they've begun to activate complement, which can kill the bacteria. In addition, there's a C3b molecule here, which is an opsonin, and then over here we've got a macrophage with an Fc receptor, and it's binding to the Fc portion of this IgG antibody, and that allows it to recognize the bacteria and mark it for phagocytosis. And another high yield thing to know about this whole process is if you have a condition where you lose antibodies or B cells or complement, you will have problems with recurrent encapsulated bacterial infections. As I discussed in some of the immunology modules, there are certain conditions where antibodies are not produced effectively or where the complement system doesn't work, and encapsulated bacteria are the major pathogens that thrive in these situations. Another group of patients who are at higher risk of infection from encapsulated bacteria are patients who lack a spleen for any reason. These patients have a risk of sepsis from encapsulated bacteria, and that's because they've lost the splenic phagocytes, which consume these bacteria after they've been tagged with antibodies. So patients who have had their spleen removed, or patients with sickle cell anemia, for example, who are functionally asplenic, are all at increased risk of encapsulated bacterial infections. You should know that the capsule is the basis of many vaccines that protect us from bacterial infection. So what we do is we use those polysaccharides in the capsule to trigger an immune response, and then we become protected from subsequent infection. Now, one problem with this is that the polysaccharides by themselves often generate a very weak immunologic response. So what is done is the polysaccharides are conjugated to some other protein that produces a more vigorous immune response. Diphtheria toxoid is often used. A toxoid is an inactive or less active version of a toxin. But some of these other molecules are often used as well, like tetanus toxoid or the outer protein from meningococcus, or sometimes a mutant diphtheria protein. But all of these proteins are conjugated to the polysaccharide so that you get a more vigorous immune response. And we use conjugated vaccine for several important bacteria. If you vaccinate someone against Neisseria meningitidis, you're using a capsular vaccine. When you immunize elderly patients against strep pneumonia, you use a capsular vaccine. And the vaccine we use for children against Haemophilus influenza type B is also a capsular vaccine. Now, some bacteria don't have a capsule, but they have a related structure called a glycocalyx. This is a sugar coat made of polysaccharides that's similar to the capsule. And down here is a highly magnified picture of a cell with these little sticky, wispy things out here, and this is the glycocalyx. So what's the difference between a capsule and a glycocalyx? Well, bacteria that have a distinct, firmly attached gelatinous layer are said to have a capsule. Bacteria that have an irregular, slimy fuzz layer have a glycocalyx. And the importance of glycocalyx is that it can be used to adhere to surfaces, especially catheters. And one of the most important bacteria that uses a glycocalyx to cause infections is Staph epidermidis. We'll talk more about this bacteria in the module on staphylococci, but it can form biofilms on catheters. So for example, if you have a central IV line in a patient which is made up of a polymer, Staph epidermidis can form a biofilm on that and cause infection. And one of the ways it's able to do this is by using a glycocalyx. Two other bacterial structures that you should be familiar with are called pili and fimbria, and these are structures that are very similar to flagella. They're little appendages or arms on the bacteria. They're made up of proteins, and they serve two important functions for the bacteria. First, they allow adherence to surfaces, and pili that adhere to surfaces are called ordinary pili. And then second, there are some specialized pili that attach to other bacteria and allow the exchange of genetic information. This is a process called conjugation, and the pili that do this are called sex pili. And there are two bacteria where it's very high yield for you to know the importance of their pili. The first one are E. coli bacteria. The pili in E. coli allow attachment to the urinary tract, and that's how E. coli bacteria cause UTIs and pyelonephritis. The second bacteria that it's important to know about is Neisseria gonorrhea. So Neisseria has pili that exhibit a feature called antigenic variation. And this means that the bacteria change the makeup of the proteins in their pili. The consequence of this is that if a patient has a gonorrhea infection, they are still at risk for subsequent gonorrhea infection because the antibodies and the immune system memory to the first infection may not apply to the second infection, since those pili will have a different structure and a different makeup. And I talk in more detail about this in the module on gram-negative bacteria and Neisseria. Some bacteria have what's called plasmids. So these are small DNA molecules inside a cell. So I've shown a picture down here with the bacterial DNA over here, but also found inside the cell are these little loops of DNA called plasmids. They're physically separated from the DNA of the bacteria, and these are important because they can replicate independently, and they often contain important genes for things like antibiotic resistance or toxins. And these plasmids can be transferred from one bacteria to another through a couple of mechanisms, and I talk more about this in the module on bacterial genetics. Another bacterial structure you should know about are the flagella or the flagellum. These are long protein arms, and this is the way bacteria move. The flagellum push them around and get them from place to place. Bacteria have their own ribosomes, and just like in all cells, ribosomes are the site of protein synthesis. And the ribosomes in bacteria are made up of two subunits, a 50S subunit and a 30S subunit. S stands for the sedimentation coefficient, or the Svedberg unit, but the bottom line is it's a marker of size. And the important thing is that these 50S and 30S subunits are different in size from the ribosomes in eukaryotic cells, and this allows us to target them with antibiotics and selectively cause toxicity of the bacteria without causing harm to the ribosomes in our own cells. And the ribosomes are the site of action of a couple of classes of antibiotics. Tetracyclines bind to the 30S unit, and aminoglycosides interfere with the 30S unit's protein synthesis, and I talk in a lot more detail about all of this in the modules on antibiotics. The last thing I'll discuss in this module is the concept of spores. So some bacteria can enter a dormant state called a spore, and bacteria that do this are called spore-forming bacteria. And when the bacteria goes into the spore form, it can survive for a long time without any food. It can survive starvation. It's also very resistant to conditions where there's not water around, dehydration, where it's hot, or where there may be chemicals. And importantly, the spore has no metabolic activity, but it can later convert to a metabolically active form. And I think of it like seeds. If you're holding a seed in your hand, you know that that seed does not have metabolic activity, but in the right conditions, it can germinate and become a living thing. Spores are the same thing. They're sort of like seeds that can turn into bacteria. So it's not enough for step one to know which bacteria form spores. You have to also know why the spores are so durable and resistant. So the outermost layer of the spore is called the coat, and the coat contains a protein that's similar to keratin, and you may know that keratin is found in our skin, hair, and nails, and it gives structure to a lot of parts of the human body. And this coat layer is impermeable to lots of chemicals and lots of antibacterial agents, and this is why it's so hard to kill the spores of some bacteria. Now inside the coat layer is a layer called the cortex, or the core wall, and this contains a lot of peptidoglycans. This is mostly a structural layer. And then one other very important substance found inside the spore is a molecule called dipicolonic acid. There are lots of these found inside of spores, and it's believed that these also help with heat resistance. So make sure you know the components of the spores. This is what allows them to survive for so long in so many difficult conditions. On this slide, I've listed some of the key spore-forming bacteria. So anthrax is a spore-forming bacteria, and that's why it's a potential weapon of bioterrorism. In theory, you could release anthrax spores into a city and cause infection in millions of people. B. cereus, which causes food poisoning, is a spore former. So is Clostridium perfringens, and tetany, and botulinum. And that concludes our module on bacteria.